Hello, everyone. Welcome back to my TCR-seq video tutorials. So one of the key features for the SC repertoire package is that it can interact with the single-cell RNA sequencing dataset. So in this video, I'm going to show you how to combine TCR-seq data with the single-cell RNA sequencing data. Then visualize the TCR clones on the UMAP of single-cell RNA sequencing data. So for the demonstration, I'm going to use the Surat full dataset. If you look at the online tutorial, the package contains a small demonstration dataset. It just contains 100 T cells. The full dataset contains nearly 13,000 cells. So the full dataset not only generates very beautiful images, but also can help you to understand the function of ESC repertoire package. So in this video, I'm going to use the full dataset. The dataset was generated from Nick's publication on communication biology. So he subset the T cells from the single-cell RNA sequencing data in this publication and used it as a full demonstration dataset. So you can download the full dataset as Surat2 RDS from this link. If you download it as Surat2 RDS object, you can update the Surat object as Surat. And also you can download the dataset as scREP full example RDS. So I downloaded this one. So we can load the full demonstration dataset and name it as Surat. So let's load it. So first we can use the dimplot to have a look at the Surat object. If we run dimplot, you can see we generated a UMAP here. The dataset has 12 cell canisters. They are T cells and named as canister 1 to class 12. And also we can use the group by function by the type to look at the cell canisters. Let's run first. We know the type separated the peripheral Bernard T cells from the tumor T cells. We can also use the table function to see how many cells in each canister. You can see here is the cell numbers in each cell canister, and the cell canister name was named according to the order of their number of cells. You see the highest C1 until the lowest C12. Because this dataset only contains the T cells, let's have a look at the dataset by clicking the Surat object. You can see here, it only contains 2,000 genes, and it has nearly 13,000 cells. So here, the dataset only contains 2,000 variable features that were selected for the data analysis. So we can use feature plot and some T cell markers to show those cells are T cells. So because we only have the variable features, here we can find the CD4 and the CD8B as T cell markers. Let's run. You can see all the cells express CD4 and CD8B, so they are T cells. So let's have a look at the Surat metadata. We can run. Then you can see the first column is the barcode. Then we have the encode RNA and features RNA for the object. And we also have the cell canisters name and the patient column, the tissue type column, and the raw barcode. Very importantly, we want to talk about the cell barcode. You can see from here the cell barcode was named as PYT. That means patients 1 tumor samples, then the barcode. So the barcode is very important to integrate the threat object with the T-cell sequencing data. That is why when we combined the TCR sequence in my first video, so we used the PY to label the first patient and the T label tumor samples, P label peripheral blood samples. In this way, we can make sure the barcode are exactly the same as the threat object. If you have different barcode, then you cannot combine your TCR sequencing data with the threat object. So we noted the threat object and also looked at the cell canisters and the barcode in the threat object. So next we can combine the threat object with the TCR sequencing dataset. So in my previous video, we generated a combined TCR sequencing dataset, and I saved the combined dataset as combined in my TCR folder. I saved it as R data. We can just click the data and load it. So now we have the combined TCR-seq data and the threat single-cell RNA sequencing data. Now we can use the combine expression function from the SC repertoire package to combine the TCR sequencing data and the threat object. So we can use the clone core as gene here for the demonstration, but you can also use AA as amino acid, NT as nucleotide, and strict as gene plus nucleotide. And we can use the group by function. I set it as a patient. Then during the combination, R will calculate the frequencies across different patient samples. So also here we can set the clone type as single, small, medium, large, and hyperexpanded. We know the highest abundance for the clone types in this dataset is 482, so we can set the hyperexpanded number as 500. So now we can combine the expression between TCR-seq and the threat object. So let's run this function to combine the data. So after combine the data, we can have a look at the metadata for the threat object again. Let's run. You can see now, after the raw barcode column, we added the barcode from the TCR-seq data, and they are exactly the same as the threat object barcode. And after that, we have the CT genes, CT nucleotide, CT amino acid, and CT strict. That's gene plus nucleotide information. And also we have the frequency for the clone type abundance. And also we created a clone type column to group the clones as single, small, medium, large, and hyperexpanded clone types. And you can see in the metadata we have a column named as the clone types, and then we can use the DIMP to have a look at the clone types using the UMAP from the threat object. We can set the group by function as the clone type, then split the samples by peripheral blood and tumor samples. Let's run. So we can zoom in. You can see now, we can see the clone type information on the UMAP. On the left-hand side are the T cells from the peripheral blood samples, and on the right-hand side are T cells from the tumor samples. You can see from the purple color, the single clones is largely presented in this area in the peripheral blood samples and the tumor samples. The small, medium, large, and hyperexpanded cells are in this area. and the hyperexpanded cell canisters is only presented in the tumor samples. So we can see the clone type information on the UMAP now. So now we can use the clone ONA function to look at the position of clonally expanded cells. So we can use the reduction of the UMAP, and you can set different values for the frequency cut point and also the bins. Here, as example, I set the frequency cut point as 30, bins as 12, and you can use the preset argument to look at any information from the column name in the thread metadata. For example, we can here set the preset as the patient. Let's run to look at the clonally expanded cells. We can zoom in. You can see we separated the UMAP by patients, PX, PY, and PZ. You can see in the PX patient, cells in this area expand their clones, and in PY patient, cells in this area and also in this area, they expanded their clones. And we know there are hyperexpanded T cell clones in the PZ patient, and they are largely in this area. Here we see it by the patients. We can also set the preset as type to see the cell expansion in peripheral blood and the tumor samples. Let's run. And we can zoom in again. You can see in the peripheral blood, T cell clones in this area expanded, but in the tumor samples, T cell clones expanded in those cell canisters. So next we can use the clone network function to look at the network interaction of clone types between different cell canisters. To use this function, we need to load the gggraph package. Let's run. Now we can use the clone network function to look at the clone type network. Again, we use the reduction method as the UMAP, and the identity will be the cell ident. Then you can use filter clones, filter identity function to filter the cells that you want to see the network. If we set the filter clones as null and filter the identity as null, we will see the network for all the clone types and all the cell types. Here we can use the clone call as amino acid to do the demonstration. Let's run. You can see we generated a clone network figure. Let's zoom in to see the whole figure. You can see because we generated a clone network for all the clone types and also for 12 cell canisters. So here the arrow color represents the relative proportion of the clones of starting node. Then the arrow shows the ending node. And the circle size here, they are in black, shows the number of unique clones in each cell canister. Because we didn't filter any clones and cell canisters, the image looks very beautiful, but it's really hard to look where the arrow starts and where the arrow stops. So we can use the filter function. For example, we can use the filter identity function to look at the clonal network just for canister 2. So we can set the filter identity as C2. Let's run this function again. Now we generated a clone network just for clone types in the canister 2. Let's zoom in again. You can see now it looks more simple to see the clone type size and see the columnar networks. We can also use the highlight column type function to look at individual columns on the UMAP. For the demonstration, we are going to use the amino acid sequence. For example, these two column types, they have the highest and the second highest abundance in this dataset. Then we can give the sequence information to highlight both columns. So let's run this function to highlight both columns. We can run first. It will generate a new column in the metadata. We can have a look at the metadata. Let's run. You can see now at the end of the metadata, we have a new column. The column name is highlight. You can see for those columns, they have the different amino acid. Then they are not labeled. We can click the highlight column. You can see. So this is the amino acid sequence has the highest frequency. Then we highlighted this amino acid sequence in our dataset, and also we highlighted the second one. Is the one with 258 frequencies. So you know now, use the highlight column function, we created a new column as the highlight. You can highlight any columns that you are interested in this dataset. So after that, we can use the dim plot function and group by the cells by the highlight column. Let's run the dim plot. And we can zoom in. You can see now this column indicated by the red color and this column indicated by the blue color. They have the highest and the second highest frequency in this dataset. You can see both columns are in the area of tumor samples that show columns have hyperexpansion. Okay, so we combined the TCR-seq data with the threat object, and I showed you how to visualize the column types on the UMAP. Look at the column type expansion, columnar network, and also highlight important column types. So I'm going to stop from here for today's demonstration. In my next video tutorial, I'm going to show you how to visualize the column types by short cell clusters. So please subscribe to my channel and share my videos with your friends. See you in my next video tutorial.